We are going to continue last class lesson. How many pages you have completed to this date? Unit number 6. 6 pages. Okay. So from there onwards, let us move. Explain Golgi complex. Good, Ashik. Good. Right. Another 2 organelles to explain you. After explaining the 2 organelles, we will go to the another portion, inshallah. Right? Here, coming to endoplasmic ribosomes. In the ribosome is a bead-like structure. Muttu madri. In the ribosome, there are 2 pieces. The large piece is called large subunit. The large piece is called by the name, how do you call it? Large subunit. And the small piece is called by the name small subunit. Actually, it is written already, but you write it by your own. How many subunits are there? 2 subunits are there. Okay. In this one, there's no any membrane outer cover. Ribosomes are found in the cytoplasm freely. Their work is producing proteins, that is protein synthesis. Protein synthesis. Every subunit is made of 2 materials. 2 materials jointly form the subunit. What are those 2 materials jointly forming the subunit? One it is protein, other one is RNA, ribonucleic acids. Are you okay? Are you okay? Good. Right. Now, endoplasmic reticulum. This is in 2 types. Look at the picture. One, it is called rough endoplasmic reticulum. And other one is called smooth endoplasmic reticulum. Rough endoplasmic reticulum, smooth endoplasmic reticulum. One is rough, one is smooth. If the surface will be feeling rough. That's why it is rough endoplasmic reticulum. If the surface is feeling smooth, that means feeling a tile, like feeling a floor tile, smooth one. Sinindu, that is smooth endoplasmic reticulum. Highlight these 2 words, take your highlight and highlight it. Okay. If you have highlighted them, we can now come to the idea regarding rough endoplasmic reticulum. These are the details. It is a flat saccade with ribosomes on the surface. On their surface, there will be ribosomes. They are flat sheet-like structures. They are flat sheet-like structures. Okay, they are flat sheet-like structures having ribosomes on the surface, right? Are you clear? And it is always covered by a single membrane, and inside the rough endoplasmic reticulum, there will be proteins. Transporting proteins within the cell is the function of rough endoplasmic reticulum. If you go for the smooth endoplasmic reticulum, it is tubular, tube-like structures, and they don't have ribosomes on the surface. They have a single membrane, only a single membrane. And what's inside is lipids and steroids. Steroids are a kind of lipids, and they transport lipids and steroids within the cell. So I have done this one as a comparison in the shape, in the membrane boundary, the number of membranes, what is the content of it, and finally, what is the function of them. Can you understand these shapes, these membrane boundary, this content, and these functions? Right. I'm trying to show you a video. In that video, you will see some more details regarding the endoplasmic reticulum. We'll try that one. Actually, we are trying that. Endoplasmic reticulum model. Let's take this one. Short video. Be cool. Can you see it? Can you see the endoplasmic reticulum? No, I have not yet shared it. That's fine. Can you see the endoplasmic reticulum? Can you see the endoplasmic reticulum? Yes. You can see. Now, my children, what kind of endoplasmic reticulum is this? Anyone? Rough or smooth? It is rough endoplasmic reticule. Rough, rough, rough. You can see here on the surface, there are ribosomes. Can you see? It's not Golgi complex. Vidurshan, where are you? Suddenly coming and telling Golgi complex. Okay, this is very Kolakumana. Okay, Golgi complex is over last 2 weeks ago, still standing on the Golgi complex. Can you see the ribosomes there? So when ribosomes are on the surface, it is called by the name rough endoplasmic reticulum. My children, these are the smooth endoplasmic reticulum. Can you get it? Tube-like. And they don't have any ribosomes on the surface. Okay? They don't have any ribosomes on the surface. Yeah, both are connected to each other, Ashik. Both are connected to each other. Two different types, but they are together. One behind the other, but they are attached to each other. Can everyone understand it? This is a model, my children. Models are good to see the 3D like view. Right. What about this? What is this? Some clay vessel. What is that? My children, it is the nucleus. Nucleus. It is the envelope of the nucleus. So what can you see there is, okay, the nucleus is connected to which endoplasmic reticulum? The nucleus is connected to which endoplasmic reticulum? Rough endoplasmic reticulum. Are you clear? Are you clear? So, once you go to the next one, once you go to the next one, vacuole. Vacuole. So vacuole is found in mainly plant cells. Okay, vacuole is found mainly in plant cells. Inside the vacuole, there will be a juice. The juice is called by the name cell sap. Inside the vacuole, there will be a juice, and that juice is called by the name okay. So if you go for the vacuole, it is a spherical structure. It is having a single membrane. The single membrane in the vacuole is called by the name tonoplast. The content inside is cell sap. Vacuole stores water. That's why it is helping to maintain balance of water in the cell. Vacuole provide support and color to the cells because in the vacuole there are pigments. and it stores water, minerals, sugars, and pigments. This is vacuoles one. Okay. Do you want to clarify anything regarding vacuole here? Do you want to clarify anything regarding vacuole here? No, it doesn't take photosynthesis. Photosynthesis is done by cell sap. Sorry, photosynthesis is done by chloroplast. Photosynthesis is done by chloroplast. Understood? Photosynthesis is done by chloroplast. It is not done by vacuole. Cell sap is the juice inside the vacuole. Sap is a juice. You know juice. Have you drunk a juice? Have you drunk a juice? In the juice, there will be water. Yes or no? Yes. There will be color. Yes or no? Color mean pigments. There will be sugar. Yes or no? No, model baba. Irunda kaata matena. Okay. So think of a juice. Now do one thing. Easy. Model is not necessary for vacuole. Vacuole is just a bowl-like structure. No need to worry about this. Is say here, this is the membrane covering the vacuole. Inside it, let's draw a don't scribble. Think this way. Right. Now this whole thing is home vacuole. In the vacuole, there is an outer cover. This outer cover in the vacuole, is called tonoplast. This inside content in the vacuole is called cell sap. Can you understand the cell sap and tonoplast? Both of these ones together, it is vacuole. Can you understand that? Both these ones together, it is vacuole. Point here is, in the cell sap is a juice. In the juice, there will be, normally the juice you are drinking, we are drinking, it has water. It has mineral ions. Mineral ions mean salts are there. Mineral ions mean what is there? Salt is there. Any juice we are drinking, are they without salt? Any juice you are drinking, are they without salt? Only sugar? Only sugar? No, no, little salt is there. Are they of coloring, pigment? So if you look at a juice, you can predict about how the cell shape is. Do you want to see a model of it? Do you want to see a model of it? Is there anything new to show it in a model? Okay. If possible, draw a diagram this way and mark it down. If possible, draw a diagram this way and mark it down. Okay. Hope all of you have completed it. If you have completed it, let's clear it. Let's go to this one. This is a model. This is the model of a plant cell. This is the vacuole, the blue color ocean light one. Then what is this? What is this? You tell. Outside one is tonoplast. Inside one is a cell sap. Right. Hope that is clear. Now, what is this? Tell will you? Now, I have explained you many things now. What is this? The purple color ball like one? Nucleus. If that is nucleus, what could be these sheet like things? Rough endoplasmic reticulum. Rough endoplasmic reticulum. If that is rough endoplasmic reticulum, what are these light blue color tube-like structures? See the place I am marking the arrow. Smooth endoplasmic reticulum. What is this? Golgi complex. What is this? Mitochondrion. What are these bead-like structures? Maybe home. Kutti, bead-like structures. Bead-like structures. Ribosomes. Right. What is this organelle, which is not much explained to you in the syllabus book in the grade 10? What is this organelle? That is the organelle called chloroplast. That is the organelle called chloroplast. The photosynthesizing organelle. What is the organelle? Type will you? Let's see the spelling. Chloroplast. Remember, this is the plasma membrane. And this is the cell wall. This is the cytoplasm. Are you clear? Right. Now it is time to do a question. Check your tute. Are you given a question there? Are you given a question there? Yes. I'll give you 5 minutes time. You complete the questions. I'll give you 5 minutes time. Until then, I will wait in this position. I'll give you. Do the questions. Okay, Ashik. Good. Put me a done mark after completing it. Don't type and send me the answer. Do it in the tute. Don't waste your time in typing the answers in the chat. I'm not expecting those things. Put me a done mark after completing it. No. Aisha, don't send. Do it. Put me a done mark after doing it. That's all. Remember, the questions are these questions, then these questions, then these questions. Up to page number 8, you are completing. By doing only a little, don't say done. Finishing up to page number 8, tell me done. You can refer, no problem. You can refer, make the answer correctly. Do it up to page number 8. Put me the done mark. Soon you complete it. Do will you, no problem. Put me the done mark after completing it. Okay. You can refer your note, no problem. You have permission to refer your note. You can come up with answers. Yeah, up to 8th page. In the 9th page, we are starting a new portion.
வணக்கங்க. நான் உங்க சமையல்காரர் பொண்ணு. இன்னைக்கு நம்ம சமையல்காரர் பொண்ணுல பார்க்க போறது சூப்பரான பரோட்டா. ரொம்ப ஈஸியா நம்ம வீட்லயே செஞ்சிடலாம். வாங்க எப்படி செய்யலாம்னு பார்க்கலாம். பரோட்டாக்கு நான் 1 கிலோவுக்கு மைதா மாவு எடுத்துருக்கிறேங்க. மைதா வந்து நல்ல பிராண்டான மைதாவும் வாங்கிக்கோணுங்க. மோஸ்ட்லி வந்து மாங்காய் மைதா அப்படிங்கிறது வந்து ரொம்ப நல்லாயிருக்கும். நான் இன்னைக்கு வந்து மாங்காய் மைதா தான் யூஸ் பண்ணியிருக்கேன். கடைகளில் வந்து மாங்காய் மைதான்னு கேட்டாங்கன்னா கிடைக்கும். நான் கிச்சனில் ஸ்லாபை க்ளீன் பண்ணி வச்சுருக்கிறேன். இதில் அந்த மாவை கொட்டிக்கலாம். மாவில் சென்டரில் கொஞ்சம் ஹோல் மாதிரி பண்ணிக்கோணுங்க. அதில் தான் நம்ம முட்டையெல்லாம் உடச்சி ஊற்ற போகிறோம். கொஞ்சம் அகலமாகவே பண்ணிக்கணும். இதில் 1 லிட்டர் அளவு தண்ணி எடுத்து வச்சுருக்கேன். பட் இவ்வளோ தண்ணி தேவைப்படாது. ஒரு 1/2 லிட்டர் தான் தேவைப்படும். மாவை பொறுத்து இருக்குங்க. இப்போ நான் கொஞ்சமாக தண்ணி ஊற்றிருக்கேன். இதில் 1 டீஸ்பூன் அளவுக்கு உப்பு போட்டுக்கலாம். 1 டீஸ்பூன் அளவுக்கு வெள்ளை சக்கரை போட்டுக்கலாம். வெள்ளை சக்கரை போட்டோன்னா உங்களுக்கு பரோட்டா நல்லா ப்ரௌன் கலரில் வரும். இதில் 2 முட்டையை உடச்சி ஊற்றிக்கலாங்க. 1 கிலோ மாவுக்கு 2 முட்டை போதும். நீங்கள் முட்டை யூஸ் பண்ண மாட்டேன்னு சொன்னால் 1 டம்ளருக்கு பால், காய்ச்சாத பால் யூஸ் பண்ணிக்கோங்க. இந்த முட்டையை மஞ்சக்கருவு நல்லா உடச்சி விட்டுறலாம்.
Nazeen, have you finished the work? Do will you? Do will you? சும்மா உட்காந்துட்டு எனக்கு போரிங் தானடாப்பா. எதையாவது பார்க்க வேணாமா? அதுக்கு ஒரு பொறுப்பு ஏதோ நல்லதை பார்த்து. How to make barotta? Wrong. Don't know, Ashik. Don't know. Quickly complete the works. No idea. Hereafter, I will indicate the rights and wrongs. Then you can find out it whether you are right, you are wrong, everything. First, do and complete it. First, do and complete it. Okay. Is there anyone still doing the works? So shall I go for the discussion? Shall I go for the discussion? Or is there anyone still doing the work? Right. Let's take the discussion into works. What is the organelle responsible for this, or structure responsible for this? Generating energy done by mitochondria. Synthesis of proteins done by ribosomes. Controlling exit and entry of ions to the cells done by plasma membrane. Allowing metabolic reactions to take place done by cytoplasm. Supporting organelles done by cytoplasm. Giving shape to the plant cells done by cell wall. Protein synthesis repeated. It's okay. Anyway, what does synthesis mean? Who will do it? Ribosome will do it. Storing cellular secretions. Storing cellular secretion is done by Golgi complex. Put your rights. Put your rights. Are you okay? Have you corrected it? Samas, it's your cute, your work. Edhula correct panna enna? Pen or pencil is not your, it's your problem, man. Right. Coming to this one. Transport of steroids. Smooth endoplasmic reticulum. Transport of proteins. Rough endoplasmic reticulum. Transmitting genetic characters. Chromosomes. Controlling activities of a cell. Nucleus. Encloses a cell. Plasma membrane. Water balance. Vacuole. Water balance goes to vacuole. Storing pigments. That also goes to vacuole. Clear? Are you okay? Are you okay? Right. So finishing that portion, we come to this. Write the organelle or cell structure that has the following feature. Matrix is found in the mitochondria. Ribosomes on the cell surface is found in rough endoplasmic reticulum. Made of two units, that is ribosome, large subunit, small subunits, ribosome. No membrane boundary, not covered by membrane. not covered by membrane. That is also done by ribosome. Rod shape, mitochondria or mitochondria contains nucleolus. Nucleolus is present inside the nucleus. Structure swollen in the edges. Golgi complex. Tonoplast belongs to vacuole, described as the powerhouse of the cell, that is mitochondria. Complete it. You can maybe correct it. Put me a done mark after correcting it. Okay. Phospholipid, plasma membrane. 10th one, phospholipid. Phospholipid is plasma membrane. Mention the organelles that are bounded with two membranes. Two membranes cover is found in the nucleus, mitochondria, and chloroplast. These ones. Single membrane cover. Rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi complex, and vacuole. All these ones are outside covered by single membrane. Organelles that are in the spherical shape. One, it is vacuole. Vacuole will look like a ball. Other one is nucleus. Nucleus also will look like a ball. Are you clear? Okay. Finishing that one. These true false statements are coming. Check your answers. All organelles are covered by a membrane. Wrong. Ribosome is not covered by a membrane. Chloroplast cannot be seen in animal cells. That is true. Smooth endoplasmic reticulum contains proteins. That is wrong. Chromosomes are thread-like structures. That is true. Mitochondria has only one membrane. That is wrong. Most abundant material in the plasma membrane is protein. That is wrong. Cell wall is made of cellulose, right? The most abundant inorganic material in cytoplasm is water, right? Golgi complex is surrounded by vesicles, right? Endoplasmic reticulum is tubular, tube-like. No, it is flat sheet-like. Nucleolus is located inside the nucleus. Okay, seventh one, only cellulose is wrong. Other hemicellulose are there. Chromosomes store genetic information, right? Ribosomes are made only of proteins, wrong. Vacuole is not present in animal cells, wrong. Vacuole has cell sap, that is right. Plasma membrane gives shape to the cell, wrong. Cytoplasm is gelatinous, right. Organelles are submerged in cytoplasm, right. Complete up to this, your correction. Complete up to this, your correction. Okay. Zainam Nawaz, vacuole, small vacuoles can present in the animal cells. It's already mentioned to you. Look at the animal cell, plant cell comparison. In the animal cell, plant cell comparison, you are mentioned small vacuoles can present in the animal cells. Yes or no? Small vacuoles can present in the animal cells. Yes or no? In the animal cell, plant cell comparison, you have been mentioned. Okay. Coming to the rest of the questions. Cell wall can't be considered as an organelle. Correct. Nucleus is the powerhouse of the cell. Wrong. Both types of endoplasmic reticulum have the same function. No. Robert Hooke introduced the cell theory. No. Cells are the functional unit of life. Correct. Cells can't appear without another cell. Correct. Cytoplasm of animal cell is pushed to a side. No. Membrane of mitochondria is called envelope. No. All cells are invisible to human naked eye. No. Neurons take part in movements. No. Erythrocytes take part in the transport of oxygen in blood. Yes. Plasma membrane is attached to the cell wall in plant cell. Yes. Complete it. Send me out of 30, how many rights you get? Out of 30, how many rights in this right, wrong? How many rights you got? How many corrects? We'll see. Okay. And now we are coming to the next work. Our next work is cell growth and cell division. What is the idea? Cell growth and cell division. I'm going to share a video with you. In that video, we are going to see cell growth and cell division. First of all, just have a look on it. I don't know how much this video is supporting, but we'll see.
Have you ever looked down at your leg or your arm to find some cut and you have no idea where it came from or how you got it? So you put a bandage on it.
Not a so good one. I want you to show that, see the cell division. Let's rest we speak about later. Just an idea.
Mitosis. In this module, you will learn about mitosis.
You have to see, now all of us, we started our journey as a single cell. Now we are growing.
Mitosis.
Grown.
In this
You are still
module,
growing.
you will learn about mitosis. Mitosis is a
This lizard has lost its tail. It is growing again.
process of cell division that makes possible regeneration of body parts. To elaborate, mitosis is the separation of nuclear chromosomes into two identical daughter nuclei. It is divided into 4 key phases. Prophase.
I want a simple one. I don't want. I don't want larger ones.
The purpose of the cell cycle is to reproduce cells. So, one cell.
Can you see the cell is becoming larger?
The purpose of the cell cycle is to reproduce cells. So, one cell.
The cell is becoming larger. Yes. The cell becoming larger is called cell growth. Later we will go for the definition. At the moment, this is cell growth. After it comes to its maximum size, the cell will divide and form 2 cells.
Cell can copy itself to become 2 cells.
Now see,
Those
now how many cells are there? Now how many cells are there? 2 cells there. After growing larger to a certain size, without growing further, it will divide into 2. Okay.
2 daughter cells are identical to the original parent cell, exactly the same. Each one can complete its own cell cycle to make copies of itself until you end up with lots of cells. This is needed for growth and is how you went from being one single lonely cell to being made of trillions of cells.
What's happening? One cell grow, divide into 2. Now 2 cell come. 2 cell grow, divide into 2. Now 4 cell come. 4 cell grow, divide into 2. How many cell will come? 8. Then that grow 16, 32. Okay, double. Increasing number.
That's how you grew. We also need the cell cycle to replace worn out.
We will speak about those things later. Now, my point it is, can you understand the change happening in this picture? Can you see the cell is growing? Right. In the picture, you are given a mature cell. Mature cell means, okay, it is the cell at the maximum size. It is the cell at the, what is it? Maximum size. Mark it down. After this maximum size, what the cell will do? After this maximum size, what the cell will do? It will divide, it will split into 2. Okay. So, if you take the growth, this is how they define the growth. Growth of a cell is the irreversible increase of size or dry mass. Cell has a maximum limit to grow. Beyond that level, cell will not grow. Instead, it will divide. Can you understand that point? Can you understand those points? Can you understand these points? Right. In the cell division, cell is dividing. How it is dividing? 1 become 2, 2 become 4, 4 become 8. Correct? That way it divides. In the cell division, now say, there is a cell. In the cell, inside, there is a nucleus. Am I right? In the cell, inside, there is a nucleus. In the beginning, when the cell is dividing, now say this is the maximum size cell. In the beginning, when the cell is dividing, first of all, the nucleus will divide to 2. Can you understand it? The nucleus will divide into 2. Now, after nucleus completes the division, the cytoplasm will divide and form 2 separate components, each having a single nucleus. Are you clear? Are you clear? So first, who is dividing? Nucleus. So it is nuclear. It is nuclear division. Nuclear division. After nuclear division, who is dividing? Cytoplasm dividing. It is cytoplasmic division. Is it clear for everyone? Is it clear for everyone? Okay. So this is cytoplasm. This is nucleus. Correct? Who is in the middle? Who is in the middle? Nucleus in the middle. Okay. What is surrounding the nucleus? Cytoplasm. Okay. Nuclear division mean one nucleus becoming two nucleus. Do you agree? In the second picture, how many nucleus there? In the second picture, how many nucleus there? Two nucleus. So what has happened to the nucleus? One nucleus has made a copy of it. Now there are how many nucleus? Two nucleus there. After nucleus copied, this cytoplasm divide here. This cytoplasm divide here. Because of division, two separate cells are made. Are you clear? Are you clear? Okay. Draw this diagram. Draw this diagram in the tute, page number 9. Anyone who complete this diagram can leave the class. Anyone who complete this diagram can leave the class, inshallah, on the coming class. Okay, that is third week of March. Again, inshallah, we will assemble on the 21st of March. Those who have completed, leave will leave. After drawing, leave will leave.